All right, welcome back again. So we'll continue from where we stopped. We're looking at protein modeling and structure prediction. I am Shadrach Eze, and this is coming from Wrestler Labs. So walk through sequence alignment, look at BLAST, how to fill missing residues, how to model proteins without an existing structure, and then obviously we'll look at major advancement in the field such as AlphaFold. Um, Structural prediction using AI, and then also co-folding. So what is sequence alignment? So proteins, nucleotides, DNA, RNA, all of these are composed of sequences of either amino acids, nucleotides, or whatever. So alignment is a way of comparing it can be one, comparing two or more sequences for similarity and obviously differences. So if you have two sequences, these two nucleotide sequences, how do you compare their similarity? So if you look at these two sequences, so how many times do they match? How many times do they mismatch? And then you can calculate similarity. For example, here we are seeing it is 90%. So that's how you judge similarity between sequences. This is usually done by what is called pairwise alignment. That is, you start the two sequences from the same end, and then you align them line by line for every nucleotide or every amino acid until you get to the end, and then you compare. Okay. Now, these similarities often indicate homology. So that means two proteins, two sequences, two DNA, they evolved from the same mother, from the same father, from the same ancestral line, right? It also can also talk about conservation of function, and then also obviously conservation of structure, because if two sequences are very similar, most likely they're going to have a similar structure. Okay, so multiple sequence alignment is an addition. It's not really an addition, but it's just a step up, because here we are looking at maybe sequence alignment for tens or hundreds of different sequences. So maybe you have the whole proteome or the whole genome, and you want to see which of them are close to each other. So you're looking at hundreds of thousands of sequences that you need to align together and find differences and then similarities. So usually, for example, we have this particular protein, histone H1, for residue 120 to 180 between the human, chimpanzee, mouse, rat, and cow. So I'm perform alignment, and they'll have to find, where do they differ? Where are they similar? You can see the asterisk is for similarity. The colon is for differences. Do you get? So the colon is for differences. And then obviously you even have gaps for differences too. So you can see that some residues are conserved, some others are not conserved. So this gives us an idea about similarity between this particular protein among these species. So how do you do multiple sequence alignment actually? So there are many methods available for us. Dynamic programming method using some algorithms, database search using FASTA and BLAST. So you have the database, let's say genome, proteome, UniProt or whatever, then you get your own sequence and try to compare with multiple sequences available in the database. Then you also have multiple sequence alignment implemented in some program like Cluster and more so. So let's look at BLAST. So first of all, you express or you write your sequences in FASTA file. It's just one of the ways to write sequence. There are other file types where we can store sequences, but here usually start with a greater than sign, then you specify some words to describe the sequences. For example, this is coming from E. coli, is a protein E. coli, and then you just write the sequence as strings of letters from beginning to the end. So if you have this kind of FASTA file, then we'll go to BLAST, which is basic local alignment search tool, available in the database. If you can see it in PubChem, NCBI database, the United States, so they have it. So let's say for this E. coli protein, this first sequence, we copy the sequence and then we put it in this box. So you enter your sequence in this box, and then you now select the organisms where you want to compare this E. coli sequence with those organisms. So, for example, Homo sapiens, that's human beings. And you can also select maybe Ratus, Navigatus, and so many other organisms. Then when you run BLAST, it will compare the E. coli sequence with all the sequences available for humans or for those organisms to try to find whether there are such kind of sequences and obviously such kind of proteins available in those other organisms. So now that is for BLAST, but let's say you have two sequences available in your hand. Let's say you have a sequence of the same protein for a cow and a human being. How do you implement a local sequence alignment in your computer? So there are very many tools. Sequence alignment is implemented in so many programs, but there are a few programs that you can run on command line like cluster W, cluster omega, which has superseded the cluster W. You can run it on the command line to detect similarities and differences between your file. So, for example, here you can install cluster omega, that's cluster O, and then you first of all put in your sequences in FASTA format. Obviously, as you can see, greater than sign sig1, greater than sign sig2, sig3. Then you run it. So, what is the outcome? The outcome is something like this. It will show you the three different sequences aligned in multiple, it's called multiple sequence aligned MSA. And then you can see here places where there's conservation and then places where there are no, for example, here there is only a point mutation. So it's like only one of the residues is out. In SIG2, as you can see, others are valine, but here is alanine. But here you can see that you can also see the same thing where there is serine, other ones are alanine. So you can identify places where there is conservation and places where there are differences among your sequences. Okay, now, if you have a protein structure, and then some residues are missing. This can be from poor refinement during NMR structure generation, or during cryo-EM, or during crystallography. So this can also be a problem where those residues they have low resolution during the generation of the structure. So if you want to use these structures for modeling, for computational work, you have to fill these residues. So what are the ways that we can fill these residues? What ways are available for us to fill these residues, right? So there's a program, PDB Fixer, is available. You can use it. There is a Rosetta. All these things can model loops. They can even model the whole protein. Modella is also there. I use Modella a lot, so you can use it to fill missing residues. Some other programs like Schrodinger, some of them implemented. I think Schrodinger Prime can fill in your loops and some other. So this can be found in some proprietary program, but some of these programs are open source, which you can use. For example, here Schrodinger Prime also has it, but some other ones like Modella, PDB Fixer, Rosetta, some of these things are open source, so you can easily use them to fill your missing residue. So let's look at an example. So PDB Fixer. So you install it, just very easy. Then I have this particular protein, 9nsr.pdb. So it has a missing residue. So add residue, add atoms, so obviously. Then add hydrogens at a pH of 7, so because it will control protonation of some charged residues. Then obviously that is the end. And then I have filled in missing residues for my protein. So if you want to see more about how to use PDB Fixer, so you call PDB Fixer help. Okay, so it can also be used, it's exposed to a Python API, so you can use it on the Python API. Okay, now, if you're working with a protein and then you want to induce mutation artificially, and maybe to find out the effects of mutation on some local dynamics, global dynamics, structural dynamics, or even binding affinity of your ligand in response to such mutation. So how do you generate this mutation in your workflow? So here we have the representation. So you have the wild type with the green residue. You have the mutant type with the residue. So how do you move from white to mutant? So, so there are so many algorithms. Let's see that. So you can use camera X, the swapper command in camera X, for example, swapper 248 training. So it will change the sorry about that, the residue 248 to threonine. So whatever it was before, I think in this particular case, the protein I'm using, I think it was initially serine, so it can change it to threonine. So it can be anything, but swapper command in Chimera works. So you can also go for bimo. So mutagenesis functionality in PyMo can also replace residues for you. So it can mutate residues. So this usually, when you mutate residues like this, they're usually a conformational search to find the most adequate rotamer for that particular residue. Yeah, because if such search is not conducted, there might be clash between the residue you are introducing and other local residues. And that is also very important because there are some mutations that cannot happen in some places in a protein because the residue you are trying to bring in is a bulky residue. For example, if you are trying to mutate something like serine, which is a simple residue, simple hydrophilic residue, into something like tryptophan or into something like tyrosine, you might have clashes if you don't optimize them very well. So on the other hand, you can use some protein prediction to like AlphaFold by just mutating the sequence so that they will generate the new protein with the mutated amino acid. So that's also very, very easy since we have access to some of these open source tools we can use. Okay, now comparative modeling. If you have a sequence of a protein, but there is no known crystallographic structure, no known structure from crystallography, no known structure from NMR, no known structure from cryo-EM. How do you work with that protein? Do you give up or so obviously you can give up, but that's not what to do. So we can actually model the structure of that protein from other similar proteins, maybe from other organisms or even from the human body. Maybe there are other similar proteins in the human body that perform similar function whose structure are already known, elucidated. So we can use those structure as building block to find out the structure of this our new protein. So homology modeling predict the 3D structure of a protein based on a similar protein with known experimental structure. So this similar protein can be of the same species or different species, but the backbone is that they must be similar. Yeah, so percentage similarity is something you query about 90, 80, whatever, but they must be similar, appreciably similar. Yeah, you can have like 40%. That if you have like 30% similar, your prediction is going to be very poor. So the higher the similarity, the better for you. Okay, the assumption is that they must be similar. The sequences must be similar. So because if they are similar, they share similar 3D structures. Okay, so what do we do? So there are two popular tools. The Swiss model is available as a web interface and a modeler which you can use from command line or Python API. So Rosetta Commons also do modeling, but these two are the ones I want to present now. So how does it work in Swiss model? So you have your sequence now, which does not have a 3D structure. So you paste the sequence here. Maybe you get the sequence from UniProt, whatever. So you paste the sequence here, upload it here. So get the target sequence, upload it here, search for templates. So what searching for templates simply means is that you are looking for sequences of amino acids or whatever. Maybe you're working with DNA, to sequences that are very similar to your target sequence. So it might be from rat, it might be from cow, it might be from guinea pig, wherever you find it, chimpanzee, wherever. Do you get? So you can search for templates with this kind of similarity. So when you find one, you now perform sequence alignment to find the extent of this similarity. So usually you are going to have results rated between, let's say, 80, 90%. You can also even include cutoffs so that you only have results that are within a very high similarity. Then once you are done, you now build your model and then quality control. So once you build your model, you will be able to predict the new structure of your new sequence, even though it's not available in the PDB, right? Okay, that's how Swiss model work. I think during the practicals we're going to demonstrate this. Okay, now Modeler 2 works as a command line only Python API. So it's supposed to say Python API you can use. Now, first of all, you install via command line or download the executable for your operating system. You can download it for Windows, you can get it for Linux or whatever. So it requires three files: the alignment file, the template structure, and the Python script. So in the alignment file, you perform multiple sequence alignment of your target sequence and the sequence of the template you are going to use. So let's say, for example, I have my target sequence is, let's say, insulin polypeptide for humans. Now, but my template sequence is insulin polypeptide chain from pig. So I'll do alignment between those two files. Then I'll also get the structure of that insulin polypeptide from pig, because that is my template structure, and obviously the Python script to run the prediction. So eventually at the end of prediction, it's going to report a molecular probability density function as an objective function. So this is just a way to quantify how reliable the model it is predicting is. So Chimera can be messy. What I mean by messy is it works, right? Yeah, but it takes a lot of effort for it to work. So, but Chimera can also be integrated into UCSF Chimera or Chimera X and can be used from there, launched from there. So as you can see here, you can the Python script, you import modeler. I mean, for modeler, import everything. Then you have the alignment file, alignment.ali. Yeah, that usually the format it takes, not as a FASTA file now, but as an alignment file. Then so it will also take the template structure. Then obviously you specify chain A because it depends on the chain number of the protein you're working with. Then you say starting model of 1 and 5. So it should generate at least 5 models and then do refinement. So it's going to do refinement for like I think 4 times for each of the model to find the most accurate prediction for your use. So now, okay, that's that. So let's look at the revolution now. AlphaFold is here because it has simplified structure prediction. We remember in 2024, Nobel Prize was given to this trial, and then Hazabis and Jumper, they were involved in open AlphaFold from DeepMind. So now structure prediction is really, really very easy. Just from the primary sequence of a protein, so it can predict virtually any protein structure by just the primary sequence. Protein, DNA, whatever, it can do that. So you just have a little limitations with maybe post-translational modifications, and it also cannot model dynamics. But I think today is even predicting ensembles. So it's really very, very advanced. So how does it work? You just need to paste your sequences here. And then if you have ligand, you can also add here. If you have ions, add. If you have DNA, maybe you want to model both protein and DNA. As you can see here, you also put here. And then you run it. So at the end of the day, it will generate the structure for you and also give you some metrics which you can use to assess confidence. You get like the quality of the structure it predicted. Things like PLDDT, the predicted local distance difference test. So it measures the certainty about each residue. So each residue, it measures its own internal metrics about how certain it is on the position of that residue in that chain. So you also have IPTM, which is interface predicted template modeling score. So for example, this is usually important, let's say when you have multi-chain, for example, if you have this protein and DNA, so how aligned are the protein and DNA? So how confident it is that this protein and DNA are interacting in this way. Do you get? So that is IPTM. Then PTM is for the global prediction. So for example, for this protein now, it looks at how confident am I that this overall structure is the structure of the protein? So usually it is colored from, blue down to maybe orange. So if you have let's say blue colors, as you can see here, it's very, very confident. If you have orange color, low confidence. As you can see here, most of the color are cyan, which shows confidence around 70% or 70% and more, 70 and 90, which is really very, very good. So we can trust this structure for use in modeling. So what we just have here is the alignment that it generated before it predicted the structure. Okay, let's talk about co-folding. So what if we don't want to do docking? We're still going to talk about docking later, but what if we don't want to do docking? We want to predict the structure of a protein and a ligand together. Do you understand? So instead of bringing a ligand and putting into the 3D structure of a protein that is already known, can we predict the structure of the protein and the ligand at the same time? And then by that, we can find where exactly the ligand is binding and possibly even predict the binding affinity. So this is the idea of co-folding. So instead of predicting the structure first and doing the docking, we predict them together, hoping that the ligand is going to find its way inside the active side of the protein. We are trusting the AI, right? The artificial intelligence to perform the magic for us. So we can also do structure evaluation. You can use docking to confirm the prediction. So what if we bypass docking and predict protein structure and ligand binding at the same step? So instead of predicting protein structure and then going on to do docking, what if we do both of them at the same step? That is the whole miracle of co-folding. So there are many tools such as AlphaFold, ESM3, ESM4 rather, most of them do co-folding. Now, the only limitation with this is throughput. Yeah, because you imagine if you have 1,000 ligands to dock, it is faster to dock it with one single structure than to feed all the ligands into the co-folding machine so that it predict the structure with the ligand for all the 1,000 steps. I mean, all the 1,000 ligands. So that would take very, very crazy amount of time. You get so. I think that is the throughput is the only limitation. But I think this technology is really advancing and it is gaining traction. People are using it and it's approaching classical methods for determining binding affinity. Okay, so that is the summary of protein modeling. So with this, we've looked at sequence alignment, how to fix missing residue, how to do mutation, how to perform homology modeling, how to use AlphaFold, and so on and so forth. So these are extra references for your consumption. So in the practical, we take it up from there. Thank you for joining us.